called complete antigens or incomplete antigens or haptens, that is. And yes, good afternoon. Okay. Okay, sir. Yeah, sure. So based on the antigens, there are different kinds of antigens, in which the antigens can be classified. That is based on the chemical properties or mode of action, based on the epitope, and then that is, unideterminant, univalent, or unideterminant, multivalent, and based on their origin, that is either they are exogenous or endogenous antigens, and based on their sources, whether they are xenoantigens totally from outside, or if those antigens are being produced within the body, they are known as alloantigens or autoantigens for that matter. And according to the function, complete or incomplete. So this is all that we had seen so far regarding the antigens. Now we'll have a look at the topic, that is adjuvants. Right. So adjuvant is a substance along with this antigen. If it is used, it produces a more immune response, except apart from the antigen alone. So it is a substance that can non-specifically, it does not, it is only going to help in getting a better immune response from the system. So again, adjuvant is it enhances the immune response in immunologically immature patients, especially in the infants, wherein you do not have a proper or a completely formed immune system altogether. So you use aluminum hydroxide or you use alum, that is aluminum phosphate, as an adjuvant in human vaccines. Other commonly used adjuvant is Franz complete adjuvant. This has heat-killed Mycobacterium tuberculosis to it. And then you have alum, that is aluminium potassium sulfate. And this, along with the antigen, be it heat-killed, viruses or bacteria for that matter, along with the antigen or the protein part or the epitope part, whatever you call it basically, it helps in a better immunological response from the body. So an antigen is mixed with alum, then the salt, whatever is formed, precipitates the alum out, and this is then the injection of this alum precipitate will help in the antigen to be released into the bloodstream at a very slow rate, so that the effective time of exposure to the antigen increases. So it can be for a few days to a few weeks with the adjuvant that the antigen exposure is taking place, because of which, a better recognition of the antigen for future purposes can be seen. Alum precipitate also helps in increasing the size of the antigen, because certain times, we are looking at everything microscopically. So with the presence of an alum, which is an adjuvant, the size of the antigen has also increased. We have already seen that, if at all there is a better or a more complex antigen, or the size of the antigen is more, it can elicit a better immunological response. So this, if the size of the antigen increases with the help of alum, then it increases the phagocytosis as well. So alum and Franz adjuvants, they help in stimulating the local inflammatory response. Mast cells and basophils play a role here. Then this attracts the phagocytes and the lymphocytes. Then infiltration of the sites at the site of the adjuvant takes place, because of which a dense macrophage-rich mass of cells called as a granuloma is formed. There are certain injections, in fact, in the 5th month, in the 7th month of the children, these vaccines, it's not the BCG vaccine per se, it's this polio and MMR vaccine for that matter. Certain vaccines, they form, the kid gets, the infant for that matter, gets a fever, and there is local swelling, and there is a local granuloma type of thing at the site of the injection. This is because of the presence of the adjuvant, and this is because of the immune response and the inflammatory response that is being seen at that particular site of injection of the antigen. Because the macrophages in the granuloma are activated, this enhances activation of the T helper cells. So then elucidation of an immune response takes place. That is, a fever will come, it will get subsided, then slowly all this will subsequently lead to the better immune memory for that matter. You have again synthetic adjuvants such as the polyribonucleotides, bacterial lipopolysaccharides. There are also other adjuvants. In fact, they help in non-specific proliferation of the lymphocytes. So again, they have given the mechanism. Let me see if at all I have this particular PowerPoint mechanism of an adjuvant. Otherwise, I'll just share in a picture relating to the same. Yes, let me share my screen. Right. So again, substances that help the or enhance the immunogenicity of the antigen. With the help of antigen only, you can get to see that the time is less and the antibody concentration is less. With the help of an adjuvant along with an antigen, you can see that it elucidates a better, what do you call it, antibody concentration. So alum, that is aluminum potassium sulfate, is one, what do you call it, adjuvant in fact. And then you have the Franz water in oil adjuvants. They are again complete and incomplete. So incomplete is it has antigens in the aqueous solution, mineral oil, emulsifying agent. This surrounds the antigen. Then it is released very slowly from the site of injection. Whereas complete adjuvant has even heat-killed mycobacteria in this water in oil emulsion. Addition, you have a dipeptide, a protein component from the bacterial cell wall. This activates the macrophages. So activated macrophages are more phagocytic in nature. So this is all about the adjuvants. So when it comes to the mechanism, you get to see that increase the strength and longevity of the immune system. You can see that graph over here more or less gives you the idea basically. Then they change the physical and the chemical characteristic of the antigen. With thus, the antigen process is improved, because of which ability for the macrophages to come to the antigen is taking place. So this can be done in the following ways. That is, effective size of the immunogen is increasing. Persistence of the immunogen over there in that particular site is there. Activating cells such as macrophages and lymphocytes is taking place. Stimulation, proliferation of the lymphocytes is taking place. Then upregulation of cytokines and chemokines, and increases the antigen uptake, presentation to the antigen presenting cells, that is the dendrocytes, is also going to take place. So when it comes to the immunobiological functions of the adjuvant, they improve the immunogenicity. Again, that means the presence of or the elucidation of the immune response of the body. And so they improve the immunogenicity of the highly purified recombinant antigens. So it can be a protein or a peptide. And then they increase the innate immune, that is the first line of defense response to the antigen with the pattern recognition receptors or other accessory cells, provide physical protection to the antigens so that there is a prolonged delivery. What happens is certain times at the site itself, in case the antigen is not active, then because of which the purpose of the immune response is not served. Then it increases the capacity to cause local reactions at the injection site, that is especially during the vaccination. So that what happens is more chemokines are released, more inflammatory markers come in, more basophils and mast cells come, so that there's a localized inflammation, because of which antigen recognition takes place. So this releases even the T helper cells and the mast cells, the basophils. So your inflammatory pathway is released. And it also helps in the translocation of the antigens to the lymph nodes. Antigens, when they are taken to the lymph nodes, that is where your memory cells, BL, and your plasma cells, the T cells come into place, and then they are able to recognize it as such that it is a foreign entity altogether. That should not be there. We should elucidate an immune response against it. So this is all about the adjuvants. Let's see the epitopes right now. So epitopes, or an antigenic determinant. So this is a part of the antigen. It is the part that is recognized by the antibody or the B cell. So this is the part that is being recognized or that binds to the T cell receptor or the B cell receptor, and then that is when the antibody comes and recognizes, okay, this is an antigen. So these are known as the antigenic determinants or the epitopes. So B cells, T cells recognize different epitopes on the same antigenic molecule certain times. So it can be only one antigen, but both B cell, if at all there are two different kinds of epitope or three different kinds of epitope, then each and every epitope will be able to attach to the T cell receptors or the B cell receptors, and then that's when the B cell and the T cell comes. This is an antigen, recognizes that particular antigen as what it is, that is an antigen, and then this elucidates the immune response. Sorry. So the epitope is the part that determines the antigenicity of the antigen, or so called as the antigen determinant. So in case that it is a protein, then an epitope may involve elements of primary, secondary, tertiary, or even the quaternary structure of that particular protein. And if at all there is only one epitope present on the antigen, it is called as monovalent. Having more than one copy of the same epitope, it is known as polyvalent. So now T and B cells recognize different epitopes on the antigen. B cells recognize the soluble antigen when it is present in the membrane-bound antibody. T cell recognizes only the peptides along with when it is combined with the MHC molecules on the surface of the antigen-presenting cells or the altered cells. So T cells cannot be considered apart from their associated MHC molecules as such. So this is how it is. Basically, the T cells and B cells exhibit fundamental differences in the antigen recognition. B cells bind the antigen, which is free in the solution. Epitopes recognize that tend to be highly accessible sites on the surface of the immunogen for that matter. Whereas T cells only recognize the processed peptides associated with the major histocompatibility. B cells can recognize if it is present in the soluble form, but T cells only if it is associated with an MHC molecule, that is MHC2 complex molecule. Then that is when it is going to go call a T helper cell, and then that is going to elucidate the antigen like antibodies. So again, let us see B cell properties. Binds to soluble antigen, no MHC molecule required. determined by the nature of the antigen binding site. So surface, that is hydrophilic, flexible basically, and complex proteins containing multiple overlapping BSL epitopes also can be bound. Whereas T cell epitopes do not bind to the soluble antigen. So the antigen peptides recognized form a trimolecular complex. So you have a T cell receptor, you have an antigen, and you have an MHC molecule also involved in this. They recognize mostly proteins, but also some glycolipids, some lipids can be presented on the MHC2 molecules. Then antigen processing is required to generate the peptides that sorry. So the antigenic peptides recognized by the T cells form the trimolecular complex. Like I had already told you, a T cell receptor, MHC2 molecule, as well as the antigen. And lipids as well as the lipids are also recognized or presented. Then it is required to generate peptides which specifically interact with the MHC molecules as well. And epitopes recognized by the T cells are usually internal in nature. So they tend to recognize the peptides that are being exposed or that have been processed within the antigen presenting cells or the altered self cells. Now, we have one more small topic, that is the haptens. So haptens are molecules that react with specific, I do not have hapten over here, I'm just like having get it. You have mitogens and superantigens as well. But in the syllabus, you do not have superantigens or mitogens for that matter. So I will just you can have a look at it. I will send this to you, but that's what. It is just only for your reference purpose. So let us have a look at haptens. Haptens are molecules that react with the specific antibody, but they do not cause immune reaction by themselves. So along with another conjugate or a suitable carrier, this coupling of the hapten takes place to another large protein, which is actually called as a carrier, because of which this hapten carrier conjugate can cause an immune response. So hapten, you can actually call it an incomplete antigen. Very small molecules, they can elicit an immune response only when they are attached to the large carrier proteins. Typically, no immune response, if at all it enters into the body also. So animals immunized with such conjugate produce antibodies specific for the hapten determinant, then unaltered epitopes on the carrier protein, new epitopes formed by the combined parts of both the hapten and the carrier. So substances like penicillin, peptide hormones, steroid hormones, they all act as haptens. So conjugates of these haptens with other proteins can be used to produce antibodies that are specific to the haptens. Conjugates, these antibodies are then useful for measuring the presence of various substances in the body. So you have this home pregnancy test kit. So what happened was, first anti-hapten antibodies to determine whether a urine sample of the women contained human chorionic gonadotropin hormone. So if it is present, that is when it is a sign of pregnancy. So it was one of the original kits wherein which was formed basically using the help of the hapten carrier protein conjugate for that matter. So to summarize the antigens, you can see that the antigens are substances which induce an immune response. They can be proteins, carbohydrates, lipids, nucleic acids, anything for that matter. Then microbes have quite a number of different kind of antigens together. They are recognized by the immune system, and then They are classified based on the antigens are classified based on the chemical nature, mode of action, origin, source, the epitope, the function, and the size. All of this matters. Then different types again: bacterial antigen, viral antigen, somatic antigen, like cell wall, flagellum, you name it, they're there. Viral antigens, they have the capsid protein as well as what do you call it, the viral like a protein also makes an important antigen for that matter. Then non-microbial antigens: snake venoms, mosquito bite, pollen grain, milk, serum proteins, then some of the food proteins, they all act as antigens. So antibody molecules are those antibody molecules, if at all they enter into the bloodstream of a different entity, they act as antigens. Histocompatibility antigens, that is, organ grafts also act as antigens. And what are adjuvants? Like I told you, adjuvants enhance the immune response in immunologically immature patients, usually the children or the infants. So alum, that is aluminum hydroxide and aluminum phosphate, they are used as adjuvants. And then very small molecules, uh, which can be viewed as single antigenic determinants, are capable or are not exactly capable of getting the immune response. So these are the haptens. So they are, if they are attached to large carrier proteins in the physical form, with the help of the T lymphocytes, they help in the formation of antibodies against the haptens. So we have seen all of this. And then, we have seen even what an epitope is. An epitope is an antigenic determinant, basically the region that binds to the T cell receptor or the B cell receptor and is presented as an antigen. So this antigenic determinant is an epitope. So if it is not there, then it is not recognized as an antigen as such. So this is all there in your antigens lesson. Now we'll go and have a look at, start with the lesson antibodies, which is, what do you call it? They are either known as immunoglobulins or antibodies for that matter. We'll just have a brief look into this lesson. So what we are going to see is we'll learn the structure of the immunoglobulins, that is the antibodies, the different types of antibodies, understand the function of the antibodies, then how B cells and T cells activate these antibodies, and then what is an antigen antibody interaction. And there are certain things called as monoclonal antibodies. We will learn about it after some time. And then we are going to see how the applications of monoclonal antibodies have made things quite easy. And then what exactly are the antigen antibody interactions. So let me share my screen now. I think it's there. Right. So as you can see, antibodies or immunoglobulins. Are you able to see the screen?
Yes, ma'am.
I was added to your WhatsApp group yesterday, so hopefully I will be able to share the antigens and the antibodies and the first lesson, that is basic concepts of immunology, in hopefully by today or tomorrow I will be sending you these PowerPoints. If at all there's anything else also, I find it a little easier for you to understand the topics and all that, I'll try to send those also. So that's what. So when it comes to the introduction of antibodies, They are soluble proteins. Antibodies are proteins in nature. They are made by B lymphocytes of the plasma cells in response to exposure to non-self antigens. So an antibody or an immunoglobulin is a specialized glycoprotein produced from activated B cells, that is the plasma cells, in response to an antigen that triggered its production. So when you do serum electrophoresis, you have globulins and that is alpha, beta, gamma, and then you have the antibodies located in the gamma globulin fraction basically. So serum globulins have albumins and globulins, serum proteins basically. So of which the globulins have alpha, beta, gamma, and in this the gamma globulin fraction has the antibodies in it basically. So each antibody specifically binds to a single antigen. Then antibody-mediated immunity controls the spread of the infection by recognizing the pathogens and their products in the extracellular environment such as blood and mucus secretions. So B lymphocytes that have performed antibodies, preformed antibodies on the surface, uh, and each B cell has multiple copies of a single antibody which is specific to a single antigen. So in order to make the antibodies, B cells have to first bind to antigens through interactions with the surface antibodies. Then this antigen-antibody interaction induces B cell to ingest the antigen which has the containing the pathogen by phagocytosis. Then when the B cells are activated by the antigen, they differentiate into plasma cells. Then after having a look or after knowing the structure of the antigen, an antibody is produced against or matching the structure of the antigen. So if you see in that manner, it is all very specific. A single antigen can be bound to a single antibody which is specific to that antigen. So now you have different classes of antibodies altogether, and they are distinguished between by their amino acid sequences. See, all the antibodies are proteinaceous in nature. Proteins again are made up of amino acids, because of which the differences in the different kind of antibodies is because of the amino acids. So let us just have a look at it. So the antibodies are also called as immunoglobulins because they immunologically react with the antigen. Then antibody and the immunoglobulin are used, the term is used interchangeably, and this is around 20 to 25% of the total serum proteins is made up of immunoglobulins. And there are 5 classes of antigen, like antibody for that matter. So you have IgG, IgM, IgD, and IgE. So IgM and IgG are found in the blood. Please give me a moment. Right. So IgG and IgM are found in the blood, and the primary antibody response is because of large amounts of IgM antibodies. Secondary antibody response is because of much more larger or even larger amounts of IgG. Now IgA is a dimer and it is found in the blood, also found in high concentrations in the mucous membrane secretions. So it's found in lungs and gut as well. IgE mast cell, so it is present in the allergic reactions. So it is found attached to the mast cells, which involves in parasite immunities and allergies. IgD is primarily as a surface immunoglobulin on the beta cells or the B cells, I mean. So when it comes to the structure of the immunoglobulin, it was Porter and Edelman. They had received, they got the Nobel Prize for determining the structure of the immunoglobulin molecule. So they used myeloma, that is cancer proteins basically. So antibodies or immunoglobulin molecules are glycoproteins with few sites of attachment for the carbohydrates. So it is a Y-shaped protein, and it is surface has a shape complementary to the determinant group of the antigen at the tip of the each Y. So this is the variable region because it has two antigen binding sites. These are the two antigen binding sites. They are known as bivalent in nature. Each antibody molecule is made up of four peptide chains. Of the four polypeptide chains, you have two identical short strands called as the light chains. These are the light chains, and two long identical strands which are known as the heavy chains. Therefore, it's known as an H2L2 structure or a heterodimer. So these four chains together are held with disulfide bonds. So you have disulfide bonds here, you have two disulfide bonds here. This is a disulfide bond. So you have three disulfide bonds. Now, two major classes of light chains, that is kappa and lambda chains, are present. And in any of this immunoglobulin molecule, light chains are either both are kappa type or both are lambda type. So you're not going to have lambda and one kappa and one lambda. Now you have five different types of heavy chains. Let's see if we're gonna have a better picture. So you have five different types of heavy chains: gamma, muta, alpha, epsilon, and delta. So that is designated by the Greek letters for that matter. So the heavy chain of the antibody that is given determines the class of that antibody. As such, IgG, that is IgG has gamma type, IgA has alpha type, IgM has mu type of heavy chain, IgE has epsilon, IgD has delta. So these are the type of heavy chains that are given. And then, the light chains, like I told you, have kappa and lambda together based after Korngold and Lepari. And you have 60% of the kappa chains and 40%. But you do not have a combination of lambda and kappa together. Never. So each heavy and the light chain has two regions. One is a variable region, one is a constant region. So that is again based on the amino acid sequences of the regions that show either variable or uniform pattern among the different antibodies. So this both V and the C terminals, the V regions are the variable regions present at the tip of the Y molecules. See, these are the variable regions. They form the antigen binding site. Rest of the arm forms the constant region. So this is all the constant region. So light chains have a constant region CL about 110 amino acids long, and a variable region VL of the same size. At the N terminus, this is the carboxyl terminus, this is the N terminus, that is the amino terminus. So at the amino terminus, the variable region of the heavy chain has around 330 or 440 amino acids. In the N terminal of the light chain, you have around and the heavy chains that they come together and they form the antigen binding sites. This whole thing forms the antigen binding site. So diversity of these variable regions of both the light and the heavy chains is restricted to only these three hypervariable regions basically. And so they are known as the complementarity determining regions, or the CDRs, which are known as the hotspots. You can see a better picture in your textbook basically if you just have a look at it. As you can see, again they have just mentioned the same thing again. variable and the constant region. Variable region is around near the amino acids, having around 110 amino acids. Then represents the antigen site binding to the antibody. They are known as the complementarity determining region, so the CGRs. So there are 3 hotspots in the light and the 4 hotspots in the heavy chain. So you have something called as a paratope also, which is a site on the hypervariable region that actually makes a contact with the epitope. So this is the variable heavy chain. This is the constant heavy chain. This is the hinge region with having the disulfide bonds. So these are the constant regions. This is the variable regions. This is the constant heavy chain. This is the the variable heavy chain. So this is a heavy chain. This is a light chain. Again, heavy chain, light chain, heavy chain, and the light chain, as you can see. So hinge region. So heavy chain region, in the heavy chain region of the gamma, alpha and delta, that is IgG, IgA and IgD, the junction between CH1, that is the C heavy chain 1 and C heavy chain 2 domain has a hinge region, which is rich in proline and cysteine. And it is very flexible, allows the immunoglobulin molecules to assume different positions so that the antibodies can reach the different antigens. So again, it is sensitive to various enzymatic digestions. That is, you have papain digestion. So these antibodies also, the regions are subjected to enzymatic digestion. So you use the enzyme from the papaya. So that is known as the papain. So papain digestion cleaves the IgG at the disulfide bridges. These are the disulfide bridges. So you're going to get the 2 fragments. That is a fragment antibody antigen binding site is known as the FAB. Let me see. I'll show you that picture. So this region is known as the FAB or the antigen binding region. And this is known as the FC fragment over here. That is the fragment which is crystallizable. This is known as the FC region. This picture will be, this is the FAB region. This is the FC region. So papain digestion, when that is taken place, the disulfide bridges are cleaved or digested, because of which the FAB region is going to be the same, but the FC region comes down. So you're going to get the heavy chains and the light chains out. So again, antigen binding region is known as the FAB region. Primary function is protects the hosts. This bears a variable region involved in the interaction with the antigens. And again, valency of the antibody is number of FAB regions it possesses. So effector function is because of the FC region, which is the constant or the fragment of crystallizable. It consists of the CH2 and the CH3 domains of both the heavy chains. So antigen specificity of the molecule resides in the FAB region, and the biological function is because of the FC, that is the crystallizable fragment region. Now, to see the different kinds of antigen immunoglobulin structures, we'll just have a look at it. Based on the different types of heavy chains, we have 5 classes of immunoglobulin structure for that matter. We get IgG, IgA, IgM, IgD, and IgE. So these 5 major classes are quite different from each other when it comes to the size, in terms of size, charge, amino acid sequences, and the carbohydrate content. So again, within these classes, there are a certain distinct differences, heterogeneity. That is, in the class itself, for example, IgG is divided into subclasses. Again, this depends upon the host species and the specificity of the immunoglobulin. So let us just have a look at IgG. So they have just given a complete comparison of IgG, A, M, D, and IgE. So let me see if you can. Right. So the form is IgG exists as a monomer. IgA can exist as either a monomer or a dimer. IgM also exists as a monomer or a pentamer. So it can exist as one single antibody or five antibodies together come together and they form the pentamer structure. IgD and IgE exist as monomers. Now valency is again 2, 2 or 4, 2 or 10. So you have two antigens can be bound at the same time to the IgG. IgA 2 or 4, monomer or dimer. 2 or 10, that is you can either bind to 2 antigens or 10 antigens. Monomer is again it can bind to what you call it only 2, and IgE is only 2. Other chains, if at all, are they there? No, they are not. Like no other chains in IgG, but you have a J chain having a secretory component in IgA. There is a J chain even in IgM. No chains as such in IgD or E. Subclasses: IgG has G1, G2, G3, and G4 subclasses. IgA has A1 and A2 subclasses. No subclasses in IgM, D, and E. Molecular weight is around 150 kilodaltons. It is around 150 to 600 kilodaltons in the case of IgA and around 900 kilodaltons. So this makes IgM the heaviest antibody in fact. IgD is around 150 and IgE is around 90 kilodaltons. So the levels in the serum are also given over here. You can see that IgG is quite a bit, in fact, around 9.5 to 12.5 milligrams per milliliter. So this is the most, in fact, that is present. So total percentage of the serum immunoglobulin, you can see that it is around 75 to 85%. And the least being IgE. Again, IgE concentration increases when there is, allergic reactions. Half-life is also given. That is, the amount of time that, it can survive. That is around 23 days. Others have a lesser half-life, in fact. Daily production is also quite a bit. In fact, milligrams per kilo, if you see 34 milligrams in IgG, the rest are all very minute in comparison. Intravascular distribution also, if you see, you get to see that it is around 45%, 42, 80% IgM. So it is more of intravascular in nature, and IgD is also intravascular. The rest are present inside the blood. And sedimentation coefficient is also given. IgM has around 19 because of its high molecular weight, as you can imagine. And then complement activation is also given over here. Whether it is a classical pathway or an alternate pathway, we will be looking at it in the next class, and whether it binds to the Fc receptors of phagocytes. IgG and IgM bind. The rest don't bind. Whether there is placental transfer, IgG is transferred from the mother to the child. So maternal placental transfer is present. Only IgG antibodies can do that, not any other antibodies. And mediation of coagulation is also there. Mucosal transport is seen in IgA and IgE, and they act as markers for B cells, which ones means IgM and IgD, because they are associated with basophils, mast cells, as well as so they are having they act as markers for B cells. Heat stable, they all are heat stable except for the case of IgE. So they have given the functions over here. This is the IgG basically. So if you get to see IgG, then you get to see that it can cross the placenta, provides immunity to the fetus and the newborn. It can fix the complements for that matter, so that you have classical pathway of the complement system that is being activated. And approximately around 146 kilodalton is the molecular weight of it. Phagocytosis is also seen. G1 and G3 bind to the Fc receptors. with high affinity so that phagocytosis of the antigen is taking place. Precipitation and neutralization is going to take place because of the help of IgG, plays a major role in the toxin neutralization. So it can, because of its less molecular weight, it can diffuse into the extravascular spaces as well. It raises long time ago. Excuse me. So it is raised after a long time following the infection. So it can represent either chronic or past infection also. Coagulation can also takes place binding to the proteins of Staph aureus, that is Staphylococcus aureus. So when it comes to IgM, you can see that it is around 10% of the circulating molecular immunoglobulins. It can exist as a pentamer, as a monomer also. It has a J chain to it. And this is how it is, the J chain is present between the two monomers for that matter. Molecular weight is around 970 kilodaltons. Half-life is approximately 5 days. And it is the first class of antibody which is made by the B cells. It is largely present in the peripheral circulation and it is the principal antibody in the primary response. So, primary immunity or primary immunological response is because of the immunoglobulin M. So, it is present only in the intravascular compartments, not in the body fluids or the secretions. So, that's what 10 Fab regions and 10 valencies is seen over here. And acute infection, it is the first antibody to be produced following the infection. So represents the recent infection, the first exposure to antigens. So primary immune response antibody. And it can fix the complement pathway. Basically, it activates the classical complement pathway. We will just look into it in the next chapter for that matter. Then you have it is a multiple complement binding site. So you have 5 FC regions and 10 Fab regions. So it is present on the B cell surface. So it serves as a B cell receptor for the antigen binding. It also acts as an opsonin. That is, it binds as an antigen, which is then easily recognized and then removed. And fetal immunity, it is the first antibody that is going to be synthesized in the fetal life. And it is an indicator of the intrauterine infection. And also it helps in protection against the intravascular organisms, and it helps in the mediation of agglutination. That is around 20 times more effective in case of bacterial agglutination than in the case of IgG. Now let us have a look at IgA. 15 to 20% or 10 to 15% of the serum IgG. Second most abundant. Two subclasses IgA and A1 and A2. So you have serum and secretory IgAs. So serum IgA has Fc receptors which are expressed on the immune receptor or effector cells. So this initiates antibody-dependent cytotoxicity, degranulation of the immunoglobulin cells, and then it also has a J chain basically, and this passes through the epithelial cells where it acquires the secretory component, and it is bound by strong covalent bonds. So IgA is present in secretions such as saliva, breast milk tears, mucus, and the mucus layers of the bronchi, the urinogenitory tract, digestive tract especially. So IgA is the most highest immunoglobulin found in the colostrum, that is the first milk of the nursing mothers basically. Now coming to IgE, it is present in the least amount, that is around only 1%. Water has the shortest half-life also, and it's minimally produced daily as well, and affinity for the surface of tissue cells, so that is the mast cells. So majority of this antibody is bound by the Fc receptors on the surface of the mast cells and the base of its allergic reaction association. So it can activate a pathway which is known as alternative pathway for the complement system basically, and it has a role to play in the immunity, especially in the helminthic, that is infections. Helminth infections means the tapeworms, and the other parasitic worms that are there basically. And IgE mediated degranulation is necessary for anti-parasitic defense. They mediate the hypersensitivity reactions for hay fever, asthma, hives, and anaphylactic shock also. Now, the last one is IgD. Again, less than 1% of the total immunoglobulin found in association with IgB again, IgG again, and it is found as a membrane immunoglobulin on the surface of the B cells. So it acts as a B cell receptor along with IgM, and uh, uh, no biological effector function has been identified for IgB as such. It is just present. Now the functions, let us just have a look at it. You can just like kind of have a look at it. We have just like seen how they act as antigens because of the presence of glycoproteins. They function as potent immunogens, can elucidate the antibody response. So you have isotype, idiotype, and you have allotypes. So abnormal immunoglobulins include the Benz-Jones proteins, heavy chain diseases, and then macroglobulinemia. So this is all about the immunoglobulins as such that is given. I will see for another particular, what do you call it, PowerPoint for you to read. So when it comes to the functions of immunoglobulins, what does it do? How does it function in the first place? So it neutralization, agglutination and precipitation, complement activation or fixation, antibody dependent cell cytotoxicity, and opsonization. These are the functions of immunoglobulins. So neutralization is antibodies block that part of the bacterial cell so that the bacterial cells effect will not be there on the body. So a neutralizing antibody is an antibody which helps in the infection of the pathogen to be neutralized completely inside the system so that the pathogen is not infectious anymore to the body. Neutralizing antibodies are a part of the human response by the adaptive immune system against the viruses, intracellular bacteria, microbial toxins. So by binding specifically to the surface structures, that is the antigen on an infectious particle, and it neutralizes the neutralizing antibodies prevent the particle from interacting with the host cells that it can be that can be infected so that they do not get destroyed. Agglutination and precipitation is when this reduces the number of the infectious units that are present, because of which clumping that takes place, that is known as the agglutination. And also floccules or aggregates of the neutralized toxin will be removed by the phagocytosis. If all of the parasite for that matter or the antigens are combined together and they are present in one place, it is very easy for the phagocytosis to occur. So that is what is happening in agglutination and precipitation. Complement fixation is you have a complement cascade which is going to come into place. Inflammation reaction will be there, because of which then this attacks this induces the inflammatory reaction. Then phagocytes come to that particular reaction. Then opsonization of cells takes place, so the antigens are recognized basically, and then this again increases more amount of phagocytosis, and then because of it this bacteria and the viruses whatever are there are being lysed totally. So this is about the complement pathway or the complement fixation. Then you have the antibody dependent cell cytotoxicity, wherein natural killer cells or the null cells or the NK cells as they are called as, you have dendritic cells also coming in. They come, kill the target cells. Antigen presenting cells will come. So T helper cells, and you have your T cell receptors that come into place. Then they help in identifying the target, and then they are neutralized. Again, phagocytosis, all of this is followed. So NK cells attach to the opsonized target cells by the IgG Fc receptor. then kill by extracellular mechanism after the attachment. And this is going to be seen in the cell-mediated immunity. Then opsonization is wherein phagocytic engulfment of the microbial antigens is taking place. IgG and IgM antibodies also have a combining site for this antigen, and it is a site for the cytophilic association with phagocytes. So bacterial and viral particles are ingested with this increased efficiency over here. Now, I will be needing another 10 minutes today if it's possible, so that I will try to finish up to monoclonal antibodies. Monoclonal antibodies I will deal in the next class, that is the next week. So let me finish the B cell and the T cell activation. I will just show you the pictures so that you can understand it. So when it comes to B cell activation and what do you call it, the T cell activation, what happens is B cell activation requires an antigen. Just give me 1 minute. I'm trying to down, like show you the pictures basically. Right. So this is the B cell activation. What happens is over here, B cell proceeds by two pathways. That is T cell, T helper cell independent and T helper cell dependent outcomes. So in T helper cell independent dependent pathway, what happens is the T helper cell and the B cells, they come together in the presence of thymus dependent antigens, and then the B cell responds to the please have a look at it, over here or into your textbook, whatever you feel is more easily to understand. So a mature B cell encounters an antigen. This binds to the B cell receptor so that B cell, it is activated. Then proliferation takes place. So this B cells form a germinal center. This germinal center B cells undergo somatic hypermutation and a class switch recombination. So activated B cells differentiate into plasma cells and memory cells. And then they have a high affinity for the original antigen that is there. Then the plasma cells in the primary loci and that are outside the follicles migrate to the lymph nodes or to the bone marrow. And then they secrete IgM within 4 days of activation. Now in T independent, that is T helper cells independent activation. So what happens is, in this pathway, antigens are called as thymus independent antigens and they activate the B cells, but there is no direct participation of the T helper cells. So the thymus independent antigens are largely polymeric or multiple repeating antigenic determinants. They generate primarily IgM or IgD antibodies. They do not give rise to the memory cells. For example, the components of the bacterial cell walls, that is the lipopolysaccharides and the capsule polysaccharide components of Haemophilus influenza, they are the ones that are T independent antigens, antibodies that are coming out basically. Now, when it comes to the T cell activation, so the class switching, whatever I have been talking about, is over here. So isotype class switching that is taking place. I had downloaded quite a number of pictures basically. Now I'm not able to exactly locate that file. So that's the problem. Please bear with me. I think I'll just go with the image that is there over here. Please give me a moment. Right. I'm just going to go along with the image that is found in the here right now. It is this right. So what happens is these T cells are found in the thymus glands. They're produced only for one particular foreign particle. That is, they're specific only for one particular foreign particle. So they are again of two types. That is, one is cytotoxic T cells, and then you have the helper T cells. So once they leave the thymus, they circulate throughout the body. Then they see until they see an antigen roaming around in the body. That is when they recognize the antigen on the surface of the antigen presenting cells, and a T lymphocyte gets activated. Now the MHC complex, which is present on the surface of the antigen, as it is, what do you call it, present on the surface of the antigen presenting cells. Now the T cell receptor on both the CD4 helper cells and the CD8 cytotoxic T cells binds to the antigens, as it is receptor TCR held through the MHC complex on the surface of the APC. So this is an APC, this is the MHC complex, and this is the T lymphocyte. All of it are together found as a bound complex. Now this triggers activation of the T cells. Now T cell activation involves a clonal expansion and it triggers a cascade pathway. This results in proliferation and an effector function or the death depending upon the intensity of the TCR, that is T cell receptor, and the signal and the associated signals that are coming out. Now cytotoxic T cells that are present, they also have a co-receptor which is known as the CD8 cells on the cell surface. So CD8 partners with the T cell receptor, and along with that, with the MHC class 1 receptors also, and it acts as a bridge. So this bridge allows the cytotoxic T cells to recognize the normal cells that are infected by the pathogen. So this T cytotoxic cells recognize this infected cell that is infected with the pathogen. Then it gets activated. It starts producing molecules that kill this infected cells so that the pathogen inside the infected cell gets destroyed. Now you also have helper T cells. These helper T cells have a different receptor, that is a CD4 on their cell surface. So this partners with the T cell receptor, interacts with MHC class 2 molecules. CD8 receptor is present on cytotoxic T cells. They have CD8 receptors. They are involved with MHC class 1 molecules, and T helper cells have CD4 markers or CD4 receptors, and they are involved with MHC2 class 2 molecules of the complex. So this allows the helper T cells to recognize the pathogen peptides that have been displayed by the antigen presenting cells. So helper T cells recognize this peptide, and then they become activated and they produce cytokines. These cytokines call in more other signaling molecules as well. So this is about the T cell activation and diversity. 5 more minutes and I will be done up to monoclonal antibody. Or do you want me to continue in the next class? Antigen antibody interactions as a tool. It is just maybe another two, what do you call it, or a slide. That's all. So in antigen antibody interactions, you get to see that. I'll just like see what all there is. They combine specifically together to form either the precipitation reaction or the agglutination reactions as such. So if they occur in vitro, that is inside the cell, basically they are known as serological reactions. They occur in 3 stages. That is, in the first stage, you have antigen antibody complex formation, that is precipitation or agglutination occurs. Second, then you have the you have the third stage, that is destruction or the neutralization of the antigen. So this is because of some properties of the antibodies and the antigens. So you see, antibody affinity is one thing wherein antigens and antibodies involve non-covalent binding to the antigen, that is to the epitope or to the variable region of the both heavy and the light chains of the immunoglobulins. So this is known as an antibody affinity. These are the methods that are there. I will just see. If you want, I will send you this particular what do you call it, PowerPoint also. Anyway, just like posture. And when it comes to avidity, avidity is the strength of the bond of the formation of the antigen and the antibody complex. So this shows the overall capacity of the antibodies to determine with the multivalent antigens. So this is because of many number of epitopes involved. So if it is injected into the blood, each antigenic determinant, that is the epitope, stimulates production of a particular antibody. And if at all there is any kind of cross reactivity, so what happens is a particular antibody or a T cell receptor can react with one or two antigens. So if they have a common epitope, so this is known as a cross reactivity. Now, these applications of the antigen-antibody reactions or interactions can be seen in determination of the blood groups for transfusion. So if at all there is a slight agglutination test done, you can see for him, because blood group with A cannot be given to a person with group B. Serological ascertainment of exposure to infectious agents such as typhoid, brucellosis, typhus fever. Passive agglutination test can be done for the diagnosis of rheumatoid arthritis, which is a type of allergic hypersensitivity for that matter. Then for ELISA test, see, this is about the ELISA test that is given in your text. This is the picture for ELISA test. You have different kinds of ELISA: indirect, sandwich ELISA, competitive ELISA. So antigen antibody reaction, basically antigen is coated to the well and antibodies are added to it. So you're going to get the substrate and measuring the color intensity. This is used for testing AIDS or the that's what human immunodeficiency virus, to detect the presence or absence of proteins in the serum for any other immunodeficiency diseases. Flocculation test for antigens and antibodies. Example is VDRL test that is done. And then you have an agglutination inhibition test for your home pregnancy, basically. So all of these, the pregnancy test kits. So they all, use the principle of antigen and antibody interactions. So now monoclonal antibodies will be dealt with in the next class. Till here, I will try to send in the slides by the weekend, in fact. All right. Thank you so much. You can all leave.